Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing with you the Feb March 2025 paper of the MCQ right now. Let's get on with this. So the first question here came for magnification. The question goes in an electron micrograph, the length of a mitochondria is measured as 17.1 centimeter. So this is the image, or you can say the value of I provided to you, and the unit is in centimeter. The magnification of the electron microscope is given as 38,000. This is M provided to you. We know the formula of magnification. We rearrange it for actual. So this is the formula that we will be using. Now, to calculate the actual length of mitochondrion, simply you divide the value of I with the value of M. So this is 17.1 centimeter divided by 38,000. You get the answer 0.000545 centimeter. Now convert this centimeter to millimeter, which is very simple. You just multiply it by 10. So this becomes millimeter. Now convert this into micrometer because the answers provided to you are all in micrometer. You have to select an answer in the unit of micrometer. So how do you convert millimeter to micrometer? You multiply it by 1,000. So your answer comes out to be 4.5 micrometer, which is D. The second question is the theory of mitochondria and chloroplasts were originally free-living prokaryote. It is thought that millions of years ago, these free-living prokaryotes were taken into larger cells by endocytosis, where instead of being digested, means they were not degraded, they stayed intact, and they became functional organisms. Which features of mitochondria and chloroplasts support this theory? So we have to find out the logic behind that. How come something that got ingested or endocytized into the cell? How was it not digested? So yes, having double membranes will prevent digestion. Then since it was having circular DNA, that's a feature of a prokaryote. So that shows that these probably were prokaryotes. And mitochondrial chloroplasts can synthesize their own protein, which means they probably were living as free living organisms once. So all three statements are correct. Which would describe the function of each cell structure, lysosome, mitochondria, and smooth ER. Now, this question is very tricky because they have given the facts regarding organelles as well. So, for example, a mitochondrion, we know the fact that it is abundant at the site of active transport. But this particular statement will not qualify for the answer because it is not related to the function of the mitochondria. So you have to make sure that you select the answer that qualify for the functions of each of these three. We know the lysosome is for digestion of unwanted organelles. Having hydrolytic enzyme is a feature but not a function. Similarly, ATP synthesis for mitochondria, lipid production for smooth ER. So let's move on. Question 4, basic components are present in a typical prokaryotic cell. Cell wall is there, 70s ribosome is there. So answer is B. We know it is ribosome is absent. Question number 5. X-ray analysis fossilized cells found in rocks in central India dating back 1.6 billion years has revealed several features. The cells are up to 145 micrometer long. We know the plant cells are about 40 micrometer diameter. So this is a pretty long, very long cell. So it qualifies for being a plant cell. The cells are joined end to end to form filaments. No, this is not a feature of plants. The cells contain some internal structures. Having internal structure is not a feature of plant again. The cells are surrounded by a cell wall. Yes, this can be a feature that you can classify the organism as a plant. Which two features were taken to when taken together provide most support to the conclusion that these individuals are actually plant cells. So this is 1 and 4. The electron micrograph shows an organelle in some cells. Which organelle is this? We know it's a chloroplast. Which row shows the structures that are expected to be present in a cells that contain these organelles? We know cell wall will be present because chloroplast is in plant cell. Centriole will be absent and plasmodesmata will be present. So the answer becomes D. What could take place during hydrolysis or reaction? A glycosidic bond is broken. Yes, hydrolysis is about breaking the bond. Water molecule is produced. No, this is condensation. It gets used up. So this point 2 is wrong. A sucrose molecule is split. Yes, again, splitting of a larger molecule is like breaking down into fructose and glucose. We know that as a fact that sucrose is made up of fructose and glucose. So yes, the answer is 1 and 3. Sugars with a ring structure can also have a linear structures as shown. Just count the number of carbons. This is 6 carbons here. So 6 carbons means it's a monosaccharide hexose. So which of these could be represented? We know that ribose, deoxyribose are all 5 carbons. So only glucose can be represented this way because all other options have a 5 carbon sugar as one of the options. Which statement about triglyceride is correct? Let's move on. The question goes as which statement about triglyceride is correct? We know it's made up of glycerol and three fatty acid chains are there. Each triglyceride molecule is formed by combining three fatty acid molecule, yes, but with a glycogen, no, that's incorrect. A triglyceride molecules contain four ester bonds, no, it contains three. Triglyceride molecules form a bilayer, no. Phospholipids form a bilayer. The ratio of oxygen atoms to carbon atoms is lower for triglyceride molecule, yes, option D is correct. Let's move on to the next question. This is question number 10. Let's review what question 10 states. They say hydroxyproline is synthesized by addition of OH group to R group of the amino acid proline. So there's an amino acid proline. If you add OH, that's they are saying it becomes hydroxyproline. OH group we know is polar. It can form it should form hydrogen bond with water. This is something very predictable, which you can just decipher from reading the question. And I always recommend my students to keep on annotating when you are doing the MCQ paper, so you know what actually you actually can predict what they'll ask as you go forward along the question. So hydroxyproline is a major component of collagen and has an important role in increasing the stability of the structure. Which statement explains why addition of OH group to proline could cause increase in the stability of collagen? So the stability of collagen is being questioned here, is being asked in this question rather. So we know that the stability here will depend on the strength of collagen. So it strengthens the hydrogen bonds. Yes, there's possibility between the R groups of adjacent polypeptide chains, resulting in a tertiary structure to prevent the denaturation. Now, this is completely going nowhere because heat denaturation is not applicable to collagen because generally collagen is very strong and adding more hydrogen bond will not make it more resilient to heat. Plus, there's not going to be any relevance of tertiary structure here because they are saying the R groups of adjacent polypeptides will be joined through these bonds. It increases the number of sites available for formation of hydrogen bonds, possibly yes, within the secondary structure of collagen, resulting in more of alpha helices. We know collagen does not have alpha helices. It increases the formation of hydrogen bond, yes, between R groups and water molecule, yes, which helps in the holding the chains of collagen triple helix together. Yes, by strengthening hydrophilic interactions. Yes, perfectly fine because nothing goes against the logic here. Part D, like option D, it strengthens the quaternary structure of collagen by providing more site for hydrogen bonding between the R groups distantly separated amino acids within the same polypeptide chain. They have minced so many words here to just confuse you. Quaternary and amino acids and same polypeptide chain, like it becomes just a minced up version of so many terms that probably a student will go for this option thinking that okay, quaternary and all these are coming there. Question number 11. Plant cell walls are strengthened by cellulose molecules that are arranged in several layers. We know that those the layers in which they are arranged, they are in each layer they're in parallel, but the layer against that layer, a different layer, is in a different direction. We studied that as this is a crisscross arrangement of cellulose, a cross-ply arrangement of cellulose in which they are arranged in different directions. So bonds that hold adjacent cellulose molecules together, no, glycosidic is holding the adjacent glucose units together. The hydrogen bonds are the ones which are responsible for holding the adjacent cellulose molecules together. What about molecules being arranged in different layers? It will be arranged in different directions. Next question, question number 12. Galactogen is a storage polysaccharide in animal species. It is a branched polymer that is formed by beta galactose monomers, which comparison of galactogen with the polysaccharide correctly summarizes one similarity and one difference. Okay, so we need to recall animals have glycogen, what is the feature of glycogen, and just relate it to galactogens to find one similarity and one difference. We can predict one very clearly. Beta galactose is this is actually coming from should it be branched, should it be unbranched, and we know in animal species we have the branched glycogen. So option A, glycogen and galactogen both are branched, yes, but glycogen is not a storage polysaccharide. No, it is. That is wrong. Glycogen E. B says glycogen and galactogen both are storage polysaccharide. Okay, maybe correct. But glycogen is unbranched. No, it's branched. See, cellulose and galactogen are both branched. Yeah, it's correct for galactogen, but not for cellulose. Amylopectin and galactogen both are storage form of polysaccharide, but amylopectin is more is form of alpha, but amylopectin is formed from alpha glucose monomers. Yes, this can be a similarity as far as these two particular molecules go. Okay, which levels of protein structures are always involved in forming the active site of enzyme? Always mean that enzyme will always have that. We know the primary and the secondary and the tertiary, both all three levels are involved in formation of the active site. So they are giving primary and tertiary, secondary is not given. Quaternary may and may not be involved because an enzyme can be made from one polypeptide or more than one polypeptide. So just in case if an enzyme, like most of the enzymes are made up of a single polypeptide, so always primary, secondary, and tertiary levels will be involved in the formation of the active site. Quaternary will only be involved for those enzymes that are made from multiple polypeptides. Catalase as an enzyme that break down hydrogen peroxide into water and oxygen. Now, whenever you are attempting a question of enzyme, I always recommend my students to just mention E over whatever is the enzyme and substrate S as for the substrate and products like P over the product. It just helps you in navigating along the question. Catalase was added to the solution of hydrogen peroxide and oxygen produced was collected in gas syringe. The total volume of oxygen produced at the start of reaction was recorded every 10 seconds for 1 minute. Like time interval was kept constant for collection. The results are shown. You can see from 0 to 10, then 20, 30, 40, 50, 60. So for 1 minute, they kept on recording the amount of the volume of oxygen being collected. So in the first 10 seconds, 22 was collected. In the next, it went up to 40. So how much was collected in between these 10 and 20? You can subtract these values and find out. So if we can see that if now we know as the reaction progresses, so there is lesser and lesser oxygen being produced in the same 10 and 10 seconds. So this means that as the reaction goes forward, the reactant is being consumed, the substrate is being consumed, so the reaction slows down. What can be concluded from these results? The reaction stopped? No, we do not know. There we can't say it stopped. The highest rate of oxygen production occurred 10 seconds. No, it always occurs in the initial most moments that should be between somewhere between 0 and 10 seconds, not at 10 seconds. It took more than 20 seconds from start for the reaction to become half of the substrate to be converted to water and oxygen. There is no relevance of Vmax, no data provided to us to show that Vmax is there and half Vmax and Km, nothing is given. So C is wrong as well. The mean rate of reaction between 20 and 30 was twice as the mean reaction between 30 and 40. Yeah, this is exactly correct. Between 10 and 30, sorry, 20 and 30, the difference of 40 and 50 comes out to be 10. So 10 cm cube of oxygen was produced. In the next 10 seconds, 50 and 55, 5 cm cube of more oxygen was produced. So yes, here it's 5, here it's 10. So this qualifies as the right answer for D. Question number 15. Succinate dehydrogenase is the enzyme that catalyzes the conversion of succinate and fumarate in aerobic respiration. Malonate is a reversible inhibitor. Recall what a reversible inhibitor do. The Vmax stays the same and the Km increases. And malonate reduces the enzyme's activity by binding to the active site. Okay, malonate and succinate cannot bind to the active site at the same time. It's a reversible competitive inhibitor. Which statement describes the effect of malonate on the activity of this enzyme? In the presence of this enzyme, this particular inhibitor, Vmax can still be reached at the concentration of the substrate is increased. Yes, correct. All others are wrong. No effect on Km. That's not possible. The presence of malonate, Vmax still can be reached. The concentration of fumarate is increased. Fumarate is a product. You can increase its concentration. It will not have any effect on the rate of reaction. Malonate decreases the Km. No, it increases the Km. You learn that as a fact in your content. Now, question 16 is surface into volume ratio, something that students get often very confused with. The diagram shows the dimensions of two blocks of agar. You have to remember the formulas, how the surface to volume ratio is calculated. Whether they give you a rectangular, sorry, I mean cuboid or a cube or a sphere or a cylinder, you should know the formulas of these. The diagram has been drawn to scale. So there's one block here and another block here. The dimensions are provided. So both are cuboids. You can see the length, width, and height are different. So this is a cube, these are cuboid. The blocks of agar were stained pink with the pH indicator. Then the acid condition, this becomes colorless. The two blocks of agar were placed in the beaker at the same time. As the acid diffused into the block became colorless. What is the surface ratio of volume of ratio block that completely colorless first? Which one of them will become colorless first? So for that, you have to calculate the surface area volume ratio of both. Now there's a trick of doing this question a bit faster. What do? You just divide the values of these dimensions by 2. So for example, this 30, 50, 20, and 40 become 15, 10, and 2. these 10, 15, 8 become 5, 7.5, and 4. So the least distance that this will be required for diffusion to occur is 2 millimeter. So just crossing the 2 millimeter, the center will be reached. Whereas the in this block, 4 millimeter have to be crossed. Only then the center will be reached. So this is probably 4 centimeters. So this is how 4 centimeter will be crossed and then it will be reached. So you can pick that this should go for, you should go for a calculation that gives you an answer using this block and not this block. You can go ahead with both blocks as well. Again, this will just take a half a minute more or a minute more. You will still get the right answer. So if you are not able to grasp what I've just outlined here using half of these dimensions, you can just ignore it. Let's move on to the calculation. We calculate the surface to volume ratio for cuboid with this formula: twice into length into breadth plus twice length into height plus twice height into breadth divided by length into breadth into height. Breadth means width. So you just apply these you just put these values in their places and you get the answer: 1600 upon 2400, which becomes 0.67 upon 1. This is the answer B. Now let's suppose if you are able to grasp this concept, just use these values and apply in this formula and you will get an answer which is obviously higher. So which will be different then, not higher, which will be different. So the which becomes colored as first, the block which is having a higher surface to volume ratio. So let's move on to question number 17. The graph shows a substance. The graph shows how the rate of acid diffusion of a substance X across the cell surface membrane changed. The concentration of substance X increased. All the conditions were kept constant. Temperature was maintained at 15 degrees Celsius. So rate of acid diffusion, you can say as the concentration of X was increased, the rate of diffusion increased, and then it became parallel to the x-axis, or you can say it plateaued. So why it plateaued? Because all the channels got saturated. Now increasing the X concentration outside the cell is not increasing the rate of diffusion into the cell. Why? Because all the channels are already occupied just before till this point. Since increasing the X was increasing rate of diffusion, it shows that the channels were still being available. They were still available for movement of this particular substance. Facet diffusion, you know, it involves channel proteins. Which statement about rate of acid diffusion is correct? The rate of acid diffusion substance X at Q will increase if the temperature is increased to 20. Yes, because it will push these molecules of X through the channels faster because this will provide more kinetic energy. The rate of acid diffusion substance X will increase if concentration of ATP is provided. No, acid diffusion does not need ATP. C, the rate of diffusion of substance X at Q will increase as the concentration of substance X is increased. It will not be affecting. We can see this already. The rate of acid diffusion of substance X at P will increase if the length of time over which the rate was measured was increased. No, it will not vary according to that because the intervals will not be affecting it. Which molecule form a bilayer? Very straightforward, phospholipid. Now, next question. A nucleus in a body cell, we know they are diploid, it's 2n, of a species having 8 chromosomes. If 8 chromosomes are there, so we know there will be a DNA molecule, 1 chromosome, 1 DNA molecule, and 1 DNA molecule is made up of 2 DNA strands. How many DNA strands are present in the nucleus at the end of interphase? Now let's recall cell cycle. In interphase, we have G1, S, and G2 phase. In G1 phase, there were 8 DNA molecules. In S phase, the replication of DNA occurs and there are 16 DNA molecules. In G2, the 16 DNA molecules stay the same, and then mitosis will start. They are saying at the end of interphase, which means we'll stop here. So these 16 DNA molecules will become 32 DNA strands. The answer will be C. In this question, the photomacrograph of a plant cell in different stages of mitotic cell cycle is shown. 4 cells are labeled as number and added to identify them: 1, 2, 3, and 4. You can see in 1 the chromosomes are around the center, so this might be metaphase. Here the chromosomes have been pulled apart, so this should be anaphase. This you can see there are 2 nuclei in this cell, so this is telophase. This is prophase because you can see the chromosomes condensing. One of the numbered cells in the main stage of mitosis, spindle formation begins. So this must be prophase. One second numbered cell in the main stage of mitosis, spindle fiber shortening. This must be anaphase. Which to correctly identify the 2 cells? We have solved it. So the answer is D. 21. The percentage of chromosomal DNA present in the cell during G1 is the same in mitotic cell cycle during prophase and telophase. So at prophase and G1, all the DNA stays 100%. The DNA will not be changed because that's the total DNA content of the cell. Question 22. The statements are about two genes and their protein products that can have a role in tumor formation, that can have a role in tumor formation. So these genes actually are the proto-oncogenes. We know that when proto-oncogenes get converted to oncogenes, these become cancerous. They cannot prevent tumor anymore. So they are saying in the first grade, the protein coded by this gene, PTEN, gene prevents cell from growing and dividing too rapidly, so it controls the rate of division. So if the rate of division is very fast, so tumor can occur because the rate is occurring very fast. Error chances will be higher. The protein coded by p53 gene prevents cell from progressing through mitotic cell cycle if the cells have damaged DNA. So yes, this will stop G2, the cell cycle at G2. It will not proceed to mitosis, so the tumor cannot set in. So answer will be A, mutation can be there in either of them, and this can result in tumor. Telomerase is an enzyme that prevents the shortening of telomeres. If not present, it is not present in normal cells. We know that as a fact, but it is active and estimated of 85 to 95% of human tumor cells. This statement explains the effect of telomerase of human tumor cells. So telomerase triggers cell destruction process. Note that telomerase will increase the length of telomeres. Telomerase damage the chromosomes. No, it cannot damage the chromosome. Telomerase can helps human tumor avoid senescence. What is senescence? Cell death or cell death. So they just do not grow old. The cells do not grow old, which is usually expected the consequence of repeated cell division as the shortening of telomere is prevented. So yes, C will be the answer. Telomerase enables the human tumor cells to divide more rapidly. No, if it was occurring more rapidly, this will become even more like abnormal. So this plays no role there. 24. The Which diagram represents one base pair in DNA. The base pair provided AT with three bonds. No, AT is two bonds. So option A and option D is incorrect. CG have three. Yes, this is correct. But one of them is shown to have this in the same direction. So both DNA strands are not in the same direction. C is wrong. In this, you can see the DNA, one strand is going upward, the other strand is coming downwards. So the answer will be B. The diagram represents the process of transcription of a gene in the nucleus of an animal cell. You can see this is the double-stranded DNA. Q should be 3' to 5' because the RNA being synthesized will be from 5' to 3'. Since it's a eukaryotic cell, we know that mRNA synthesizes the primary transcript, which then undergoes the post-transcriptional modification where the exon introns things goes on and splicing, etc. occurs. So which row correctly identifies P, Q, and R? P is not an mRNA. P is a primary transcript because we are talking about an animal cell, which is a eukaryote. Q is not a transcribed strand. The Q strand is a template strand. We use the term template for it and not the transcribed strand. Similarly, the strand which is not being used, it is called non-transcribed strand. So the answer becomes D. Which row correctly describe the role of growing leaves as source or sink? Now, since these are growing, so they will behave as source as well as sinks because they are growing. At the same time, they are photosynthesizing. At the same time, they are using up the products as well. So they can become source as well as sink for both these products. Which row correctly matches each pair of description fruits like to cilia and to root hair. We can recall root hair cells. They can root hair have vacuole. You can recall the diagram even from your O level. You do draw a vacuole extended within the root hair. What about cilia? Yes, cilia are present more than one in a cell. It's never a single cilia. Cilia is like a cell, and then there are many cilia like protruding out of that cell. Question number 28. Which changes occur as amino acids are moved from phloem sieve tube to a source? We know it lowers the water potential as more and more of it gets loaded into the phloem sieve tube, and the volume of that fluid will increase. So answer becomes B. 29. The graph shows dissociation curve of hemoglobin at two different partial pressure of carbon dioxide. Which labeled point in the oxygen dissociation curve will result in the highest concentration of hemoglobinic acid? Now, hemoglobinic acid occurs in the respiring tissue. We know in the respiring tissue, PO2 will be low, they will be high, PCO2, HHB formation will be occurring faster, and chloride shift will be occurring faster as well into RBC. These are just the random facts that you can recall and annotate whenever you come across a question where hemoglobinic acid is highest being concentration being asked. So which curve do we pick? Low CO2? No, we will pick the high CO2. The answer should be either B or D. Why don't we pick D? Because D shows that oxygen is very much there in blood. So this is highly oxygenated blood. D is high oxygen. So this cannot be true. So the answer should be B because here the PO2 should be low and PCO2 should be high. Which statement about transport of carbon dioxide into blood are correct? So if you go through all these statements, except for statement 4, all are correct. These are some basic facts. Hemoglobin, the statement 4, why is it wrong? Hemoglobin readily combined with hydrogen carbonate ions. No, it never combines there. Hydrogen carbonate ion is formed by because of dissociation of the H2CO3 and it goes out into plasma and the chloride comes in. Chloride shift. Question 31. What happens as a result of blood pressure in the left ventricle becoming higher than the blood pressure in the left atrium? We know when the left ventricle pressure rises beyond that of left atrium, we know that your AV valves close, so blood should not flow back into the left atria. It should keep flowing into the aorta. Now, we can very clearly omit out B and C, but option D states the similar valve of aorta opens. We know that during left ventricular systole or ventricular systole, we know that similar valves do open. But the opening of the similar valve is due to the difference in the pressure of left ventricle and atria, not the difference in the pressure of atria and left atrium. The question here very clearly asks you about the pressure difference between the left ventricle and the left atria and the consequence of it. Whereas the opening and closure of the cellular valve is because of the pressure difference between the left ventricle and the aorta and has got nothing to do with what's happening with the atrium. The scanning electron micrograph shows inner surface of human trachea. So this is human trachea shown. We can see that must be ciliated epithelium and something other than that should be the goblet cell because these are all that's present on the surface of human trachea. So which what identifies in this thing D. So this should be goblet cell. Ciliated shouldn't be the answer because these are the ciliated ones. So goblet cell should be the correct answer. Mucus should not be correct answer because mucus are these tiny things, these individual structures being released. These are mucus. The whole structure T is a cell that produces mucus, so that should be goblet cell. Which row correctly summarizes the typical distribution of cartilage and smooth muscle? We know cartilage is present in trachea and bronchi, where smooth muscle is present in trachea, bronchi, bronchiole. So the answer has become 1 and 3. Bronchi is having both cartilage and smooth muscle. Trachea is having both. Bronchioles is not having cartilage, yes, but it is having smooth muscle. That's an incorrect thing. So 2 is wrong. A molecule of oxygen diffuses from the alveolar air, from the air in the alveolus to the hemoglobin in the red blood cells. So if it's coming in this direction, so if you can see if it's crossing all this way, so how many membranes will it be crossing? So you can say minimum number of membranes. So we can see here that inside the alveoli, you will have what? A molecule of oxygen. You have to move this molecule of oxygen across the endothelial cell of the alveoli. So that takes up two cell membranes. Then it will cross the epithelial, or you can say endothelial cells of the capillary. So again, two membranes will be crossed there. Then the RBC membrane will be crossed, which is another membrane. So a total of 5 membranes have been crossed. You multiply 5 by 2. So that gives you the answer 10 because every membrane is a phospholipid bilayer. They're asking you about the number of phospholipid bilayers. Question 35. Which shows the type of pathogen that causes cholera and its mode of transmission. Cholera is caused by Vibrio cholerae, which is a bacterium. So A and B cannot be the answer. It is not by airborne droplets, contaminated water. 36. This statement of tuberculosis is not correct. TB can be controlled by vaccination. Yes, it can be. TB is caused by a virus. No, it's caused by a bacteria. So since this is, we have to find out the fact which is wrong here. So answer is B. All other facts are actually what actually is going on with TB. 37. More cases of malaria are being reported in Europe. Other diseases formerly confined to tropical countries are transmitted in the same way as malaria have also spread to parts of Europe. Tropical countries have higher mean temperature and humidity than Europe. So what's happening? That global warming and all is occurring, rising temperatures, humidity cause more mosquitoes to keep on breeding. There's climate change occurring. Yes, that will cause more mosquitoes to breed. Increase in travel, yes, will cause more people carrying the disease to Europe. Then creation of wetlands will provide marshes. And although this is done to increase biodiversity, but this again provide more breeding ground for mosquitoes to be there. So all three options are correct. The diagram shows 3 different bonds. Which bonds are likely to be there in an antibody molecule. Identify the bonds first. So this is a hydrogen bond shown by a dashed line. This is a peptide bond. This one is a peptide bond, and this one is a disulfide bond. So an antibody molecule is a glycoprotein. We know it is having hydrogen bonds and glycoprotein, and and it's having the peptide bond and disulfide. So all 1, 2, 3 are correct. Specific monoclonal antibodies can be used to treat some forms of tumor. One example is trastuzumab, which is used to treatment in the treatment of tumors caused by cancer. Which treatment could help to explain why monoclonal antibodies such as trastuzumab are suitable for this role? The variability of monoclonal antibody trastuzumab can change. No, the it cannot change. It should not change. So option three is correct. Option one is wrong. Monoclonal antibodies such as trastuzumab bind to specific targets on the cell surface receptor of cancer cell. Yes, which prevents the receptor from giving signals that are needed for cancer cells to grow and divide. Yes, that can be correct. So the option becomes 2 and 3. HIV virus cause illness by infecting and destroying helper T cells. When helper T cells get reduced, we know cytokine level drop down as well. This leads to agent develop an ability to produce an effective immune response. Which components of immune response are produced less effectively when AIDS develop? So all these three are affected. The memory cells deplete, the plasma cells cannot form, antibodies production stop, all because the cytokine levels have dropped. So all option 1, 2, 3 are correct. So that's it from my side. I hope this was helpful for you. Thank you so much.